Hi, everyone. It's my pleasure today to introduce Justas. Justas is a postdoc at the Baker Lab at the University of Washington. He obtained his bachelor's and master's in mathematics and his PhD in biophysics, all from the University of Cambridge. Today, Justas will talk about his work on ProteinMPNN. I'm sure all of us have seen the work recently come out in Science, and we're all very excited to hear what you've got for us. So I think we're going to keep questions for the end, but feel free to put them in the chat in the meantime. Take it away, Justas.
Thank you for the introduction. So I'll be presenting on ProteinMPNN, and please note down the questions and then ask them at the end. So first of all, I would like to thank the whole ProteinMPNN team. So there's lots of people who have been working on it, from both creating database, training models, and also doing experimental work. And usually when I present the work, I go into the details, how the project started, and present all the sort of mathsy bits, but I'm just going to reverse and explain what is ProteinMPNN and present the experimental data first, and then we will dive into some of the metrics. So the ProteinMPNN is the model to map protein backbone geometry into protein sequences. So this structure to sequence and sequence to structure mapping, and we all know, heard of AlphaFold and Rosetta fold, predicting from a sequence structure. So some of the experimental results, one of the papers that came out at the same time with ProteinMPNN is called Hallucinating Symmetric Protein Assemblies. And in this case, this is a figure from the paper. So Lucas, Basil, and Alexei in our lab, they've been playing with hallucinating or generating proteins using AlphaFold. So in this case, they're interested in a cyclic protein. So in this case, for example, 3 copies of the protein of exactly the same sequence are introduced initially, and then we can use AlphaFold to predict the structure and impose some losses. So for example, ask the model to be very certain, so high PLDT and PTM, but also introduce the cycling loss, and then do Markov chain Monte Carlo, MCMC, to try to mutate some sequences and keep doing this loop until we get really the metrics like we want. And then at the end, I will show that these designs needed to be redesigned with ProteinMPNN, some sort of model that maps structure to sequence, because the original design did not work. And if we look at the sort of the solubility, so the how much soluble yield there was from different designs. So this original Markov chain Monte Carlo hallucination for these homo-oligomers, those were the solubility results. So on, I guess the median is about 10 milligrams per liter, but then, so a bunch of them are really low yields. So they sort of say insoluble proteins. They aggregated, redesigning the same exact backbones of ProteinMPNN increased the soluble yield by quite a lot. This is a log scale. So now we're talking about hundreds, even close to thousands of milligrams per liter. Also from these oligomeric designs, we got crystal structures. So this showing 7 crystal structures, some of the homodimers and trimers and so on. And the gray are the models, and the solved structures are colored. And there's some interesting designs that the beta sheets and alpha helices wrapping around. So that's quite exciting. And they all match quite well the design structures. I guess I should say that all of these were, so I'll probably mention later, but the redesigned sequence of ProteinMPNN, and again, are repredicted by AlphaFold to make sure that there's a match between what we try to design and what we redesign with ProteinMPNN. Also, we can hallucinate these like large structures. So these different rings of different symmetries of C18, 6. So there's a 6 units, and there's internal symmetry of 18, and this showing a cryoEM and negative stain microscopy results. So that's quite exciting. There's lots of big structures, and I'll show ProteinMPNN can redesign these symmetric proteins, making sure that the sequence stays symmetric. In the Institute for Protein Design. We also have the King's Lab. They're working on different nanoparticles. So in this case, Robert D. Haas redesigned some of the tetrahedral nanoparticles. So in this case, the original design was done with Rosetta, fast design, and the interface was not forming, this particle was not forming together, but then redesigned the ProteinMPNN, quite similar backbones that failed initially from earlier papers, now give really nice crystal structures that match, and these particles are really stable. So that's quite exciting. One of the other examples is that Huabei in our lab was trying to design, incorporating these short linear motifs into de novo design protein scaffolds. So the idea is that, for example, for this SH2 domain is to bind this native peptide. So we want to come up with a protein structure, this de novo small peptide that would support this green structure. So originally it was designed with Rosetta remodel, layer design, and packing, but there was no binding signal. But then redesigning the ProteinMPNN whole this orange structure, keeping the green structure as the given native, so trying to support it, provided binding, and then just to test whether some of these residues are actually supporting the structure, there were modifications made from asparagine to aspartate, and it indeed was supporting this small green structure. There are more papers coming out that are using ProteinMPNN. So this is one example from Nate Bennett. It's called Improving de novo Protein Design using Deep Learning. So some of the failure modes for designing proteins is that if we're trying to design a binder for a target, sometimes the binder itself doesn't form. So it's type of 1 failure, but sometimes also it doesn't bind. So it's type of 2 failure. And there's some examples sort of showing metrics. In this case, the metric is AlphaFold success of the predicted align error using different methods like Rosetta design, ProteinMPNN, and also people try to run ProteinMPNN and run fast relax to move the backbone a little bit, and then run ProteinMPNN again. And you can see that the third method of running ProteinMPNN and fast relax in cycles is increasing this AlphaFold success rate, at least sort of tricking AlphaFold to think that those are good binders. And also in this paper, there's some benchmarking results showing that with the previous results, AlphaFold predicted align error can be used to discriminate better binders from the worst binders. Another paper that used ProteinMPNN in our lab is related to the hallucinating these symmetric oligomers, but in this case, thinking about creating proteins with pockets. So these pockets would be used for binding different small molecules. So the same idea of designing these oligomers, but then redesigning the ProteinMPNN and removing the symmetry. So allowing these 3 units to be of different sequence. So the sequence would become different. So they wouldn't be perfect homo-oligomers, but they would have different chains. But they call it pseudo-symmetric. So the backbone is symmetric, but the sequence is not symmetric. So it's always, I guess, fun to do a little demonstration. So I'm going to show this Hugging Face created by Simon and try to predict this this top 7. So this is de novo design protein 1QRS. This figure was made for the IPD, so Institute for Protein Design, celebrating 10 years. So we can try to redesign this sequence and then predict with AlphaFold. So this is this Hugging Face setup. There's also a GitHub code where it has maybe slightly more options how to make, but this is a very simple setup. So in this case, we can either upload a PDB or write the code. So 1QIS was the code. And if we go to the settings, we can choose which sequence to design. So this design, there's only one chain let's say design 4 sequences. Use the sampling temperature. Let's go smaller sampling temperature. We can choose what sort of model you use, and I'll talk about these different models trained with different amount of noise. So let's use a default, and then we can just run the model and design sequences. So it is quite fast. That's it. This was the original sequence, and then now we redesigned 4 sequences, and the model outputs the temperature they were designed at. What is the score? So this is the negative log probability. So lower the better. Then the sequence recovery. What is the match between the output sequence and the input sequence? What was the model name? So we have about 40-45% sequence similarity from the input native crystal structure to the design sequences. And now we can do structural validation. So we're going to run AlphaFold with 3 recycles on all of the sequences. and it's going to take about 30 seconds. So I'll go back to this later to see what is the match between the input and the output. So if we continue, I'll share my slide. 1 second. Okay, so while this prediction is running, I'm going to continue describing the model. So we have a structure to sequence model, and some of the analogies that often people think in protein machine learning are these sort of, I guess, computer-related image generation or text. So in this case, I'm sort of showing this task of mapping from protein backbone into the sequence, and then from sequence into the backbone as an analogy between mapping an image, describing it as in a caption. So in this case, this is a small cactus wearing a straw hat and a neon sunglasses in the Sahara Desert, and then using the text to generate the image. So in this case, this image was generated by ImageN, giving this text prompt and generating the image. So this is sort of like a generative model that will produce many solutions. In this case, this ProteinMPNN is more like mapping an image into the text, but the distributions are quite different. In this case, it's a 2D grid of pixels. In a protein world, it's 3D coordinates of the atoms. And from this sort of like a translation perspective, the number of residues are matching in the coordinates and also in the residues. So we have a matching length, and there's only 20 letters available. So a somewhat simpler maybe task. The task of predicting structure is quite difficult and it requires homology and other information. So the problem statement, what sort of was the idea behind ProteinMPNN, is to come up with a model that can sample sequences that are highly likely. We could model multiple chains, we could fix parts of the chain, and we provide uncertainty about the samples. So in this case, this could be an example of 2 chains, 1 chain here and 1 chain here. And we want to maybe fix everything and just redesign the interface. So just redesign the red parts, giving all the rest. And the model would like to output the sequence and also the probabilities of the sequence.
What are you giving him? That one you gave, the one at 9 a.m. was 42, and then you added more.
So the examples would be one-sided binder design, homo-oligomer design, enzyme design, and so on. The training data was collected by Ivan Anishenko, so it's a similar training data for, I guess, Rosetta fold, but in this case, we're using the PDB bi-units. So these bi-units, we collect the single chains of the PDB, clustered at 30% sequence identity. We're using 3.5 angstrom resolution cutoff, and taking the complexes that are smaller than 10,000 residues. And so in the training data set, there would be a bunch of homo-dimers, homo-oligomers, and we are asking for the model to only predict one single chain that we clustered in the context of everything else. So it would be a very simple task if we asked, for example, given the blue chain that's exactly matching the red chain to predict the sequence, that would be, the model can just copy over. So what we do, we check the sequence similarity to see if it's more than 70% similar to from 2 chains. And if they are, then the model has to predict sequence of all the chains instead of only 1 chain. So preventing this data leakage from the homo-oligomeric and similar sequences. And the objective for the model is to minimize the categorical cross entropy. So this P distribution is the original distribution. So in this case, for example, it's a glutamate. So it's a probability of 1. This is a native distribution. And the model is outputting a distribution Q. That would be some distribution that the model thinks it is. And then we would just compute the log probability of the correct amino acids, so glutamate, and sum them over. So in this case, the model is using autoregressive decomposition. So for example, given a small bit of the sequence that is already decoded and the backbone, the model produces the probabilities of the amino acids, and then we sample from this distribution to get a sample. So in this case, it's sampling E, and then using this information, we predict again a new probability distribution for the next amino acid, and then again samples. So in this case, it's proline. So the model is multi-step, or autoregressive is decomposed, that one amino acid at a time, it is predicting distributions, and then we sample from these distributions. So just comparing this left to right decoding that is more common in a language translation, and ProteinMPNN was using arbitrary decoding. So doing training, a random decoding order was given, and the model had to learn all different decoding orders. So in this case, for left-right decoding, we have some text that's on the left, and we just predict the next as a function of everything on the left. In the arbitrary decoding, we can, for example, still want to decode these two amino acids. The model can use both the context on the left and on the right, whereas the left-to-right decoding would not be able to use, for example, these three letters to the right. The input features to the model, so talking about these geometric features, are, first of all, we sort of assume that the local context will be the most important. So we're forming these local neighborhoods for every residue in terms of the C-alpha distance. And then the input features to tell that residue I and J are, for example, in different chains. So the green nodes or these circles are chain A, the red one is chain B. So we would give the plus minus 32 in the primary sequence. So for example, this residue would know it's one residue to the left of this other residue, or two residues to the left of this residue. If the residues are in different chains, then we just give a sort of binary indicator, say that these residues are in different chains. So this is similar to the AlphaFold encoding of the positional encoding, but plus also taking care that it doesn't matter whether chain A is called A or called B, so that there's no A goes before B. So it's just an indication whether it's the same chain or different chains. And then on the edges, we also have the distances. So we have these radial basis functions, I'm not sure what they are, for distances between backbone atoms. So N, C alpha, C, and oxygen. And there's also a virtual C beta that is calculated as a function of the other residues. To test whether this assumption of the local neighborhood is correct, we can train neural networks and check sequence recovery as a function of the neighbors in the graph. So this is, for example, using 16 nearest neighbors, 32 nearest neighbors, 64 nearest neighbors, and we can see that the performance in terms of sequence recovery is converging. It's not getting better if we make the graph more and more connected. So it is mostly the local information that is important to predict amino acid identity. So these distances, so these input features, these 25 distances between N, C alpha, C and virtual C beta. So in this case, this is N, C alpha, C, oxygen, and C beta. So we give all the interdistances that are encoded as like radial basis functions. So we can imagine these like little channels that are exponential of the true distance minus these middle of the distances of these bins divided by some standard deviation. So they go only from 0 to 1. Everything is very nicely normalized, and if the distance is really big, it will just get everything with zeros, and if the distance is very small, there'll be maybe a very small blip for the first bin. We try to do all the different variations with angles, so giving dihedral angles as an input, relative frame angles, and so on. It really does not work as well, or doesn't add on top of what the distances give. So distances are probably a better inductive bias. Maybe it's easier for the model to learn what's going on. So just giving distances between all the atoms. So things we have so far is we made this graph that is a local graph, and we have features on the edges that are this positional encoding and also these geometric features. And then we want to predict these probability distributions per node given the rest of the amino acids. So amino acids will be encoded as nodes, that some of them that are decoded. And then we have the true, we can calculate the Cadwell cross entropy loss. Since we have to run this model as many times as there are amino acids, it is useful to somehow discretize the model. So basically you have a backbone encoder and a sequence decoder, and that's very often done in the language translation to having an encoder and decoder. So the encoder job will be to encode the backbone, and then the decoder job will be to run iteratively multiple times to decode one amino acid at a time. So let's dive in slightly deeper, a little bit deeper into the backbone encoder. So the backbone encoder, as we're saying, takes these distances between different atoms as node features, and then there are no as edge features, and there are no node features. So the model is having these edges and nodes. Nodes are just initially 0, so it can just be omitted. And it's a message passing neural network that updates the nodes and then updates the edges to come up with better nodes and better edges. There are 3 layers of those that can be run. And then the decoder takes the final encoded edges and nodes and also adds the sequence into the nodes, the one that was already decoded, to produce probabilities from which can be sampled during training. We're not sampling, we're using teacher forcing to just run in one pass all the decoder. But during the inference, we can sample one at a time and run decoder multiple times. So the model architecture is really lightweight. It's about 1.7 million parameters. So just 3 decoder layers, 3 encoder layers, 3 decoder layers. The hidden dimension is 128, comparing with AlphaFold, which has about 100 million parameters. So this is a really cheap to run even on CPUs, and can run on really big proteins like 10,000 of residues or more. One of the important aspects in training these models is adding a backbone noise. So since the crystal structures probably have some artifacts, they're very, especially the higher resolution ones. And when people are designing things, we're really not sure about the backbone. How does the backbone exactly is positioned? So we want to make the model that is not very sensitive to the input coordinates. There's some slack. So we're adding Gaussian noise during training to all the coordinates. But then during the inference, there's no need to add any noise because the model is already, for example, not paying attention to the scales of 0.2 angstroms. So this is one of the benchmarks. So the whole idea for the paper was born from the John Ingram's paper, Generative Models for Graph-Based Protein Design. And we try to build on top to see what could be improved. So this is a table showing that if we take exactly the model from John Ingram's method, and then we just change one thing at a time. So this model is not going to exactly match the model that I just described here, because in this case, there was some dihedral input features and so on. But if we take the baseline model that is exactly in this paper, and then we just change one thing at a time. So experiment 1 is adding these distances as an input features. We can see that the sequence recovery straight away goes from 41 to 49%. So there's definitely adding the interdistance feature was a really helpful inductive bias. And then updating encoder edges in the normal sort of vanilla MPNN is only node updates, there's no edge updates, is giving a small increase compared to the baseline. And then if we combine 1 and 2, we get a slightly higher boost. And then if these all, the baseline experiment 1 to 3 are all left to right decoding autoregressive models. If we switch from the left to right to a random decoding order, so the model has to learn all the possible decoding orders, there's no decrease in performance. It's almost the same. And one interesting thing to notice is that this noise level during training is that the original models were run without any noise. And we also test what would happen if we add a very small 0.02 angstrom standard deviation Gaussian noise. And you can see that the performance is on the PDB crystal structure is getting lower. So even at the highest, it's about 3% difference. So it means about 3% of the performance is coming from this really, really tiny amount of distance that the model is either picking up on the crystals, that maybe the distance between C alpha and C alpha is indicating that it's going to be specific amino acid. But if we take AlphaFold models that are idealized backbone, the difference is much smaller, or sometimes it's even is better to train with model that is robust. It's not overfitted to the crystal structure. So that's an interesting observation. All of the other results that I'll show will be for the model that is actually ProteinMPNN on the GitHub. So this is the only one that is more like a toy example. So comparing the Rosetta that's been mainly used in a Baker lab, I'm plotting the sequence recovery as a function of the average C beta distance. So I wanted some measure that is independent of the side chain packing that would show how buried the residue is. So we have a core residues that are in the core of the protein and then the surface residues. And I'm showing the fraction of the residues that fall within these distances for the nearest C beta. And this is this virtual C beta. Even if the atom doesn't have C beta, we calculate virtual C beta. That on average, there, for example, 30% of residues at this 6.2 angstrom 8 neighbor closest distance. We see that ProteinMPNN is doing better than Rosetta in both in the core and on the surface. And they're both kind of matching at the core. There's a really high certainty with almost 90% accuracy or sequence recovery. The model can recover what was actually there. And then on the surface, it's much lower, but they're both almost monotonic functions of this nearest C beta distance, which is an interesting, suggests that the the ability to recover the sequence it's a function of the geometry. It's saying how much of the constraints I have from other residues around. This is not incorporating any function or any other things. If we look at the protein by protein basis, so one dot is one protein. I think this is about 400 monomeric structures. ProteinMPNN sequence recovery is better than Rosetta in most of the cases, except this one point. And there's definitely a correlation. So this correlation is probably coming from intrinsically how well packed the backbone is. We could also look at the sequence recovery. So the model was trained on complexes, has been trained on homomers, heteromers, and interfaces. So we can plot out how it performs the sequence recovery for these different groups. So when using the model, there's this sampling temperature. So I'm going to just quickly explain what the sampling temperature is. So what it mainly does, it makes the amino acid distribution sharper. So ProteinMPNN takes the backbone as an input and then produces these called logits. So logits are the final outputs of the neural network before using any other things. And probability distribution is formed as this exponentiation of the logits divided by this temperature. So it can adjust the relative scaling between the logits. Of course, it's normalized, so it adds up to 1. So making the temperature small makes the distribution very sharp. And if we increase the temperature, we make it almost uniform. We can think at training time, the temperature is set to 1, according to the laws. So we can produce a higher probability sequences, probably by getting some bias by reducing this temperature. And I'll show how this temperature affects different things. So for example, sequence recovery as a function of the temperature is for these monomers and homomers is slowly decreasing. So we're sampling less and less matching sequences to the input, but then the sequence diversity, if we try to design multiple sequences and see how similar the sequences are, the diversity is increasing. It's interesting to notice that at 0 temperature, so it means taking argmax, the sequence diversity is still not exactly 0. It's about 10%. And this 10% sequence diversity is coming from this random decoding order, that these models are slightly different from each other. And if we look at the sequence similarity per this burial, look at the core versus surface, so different temperatures, we see that the core is quite the same, but then the surface is quite different. The sequence similarity is about 75% for the surface residues at the low temperature, and only about 47% at higher temperature, which means there's lots of uncertainty and the probability distributions are quite wide on the surface. The model is not sure what amino acid should go on the surface, but the core is very well resolved. If we look at the amino acid biases, so in this case, I'm plotting the model's bias minus the PDB bias. So we can see the things that are above, it means they are overrepresented, and the things below would be underrepresented. So if we look at as a function of the temperature, the lower the temperature, the higher there is the bias. So at 0.1, there's more negatively charged glutamates. If we look at the temperature 1, which the model was trained, it's matching quite well. So there's only a really, really small bias with respect to the true distribution. But this bias is quite interesting, that the model is trying to put more glutamates and lysines on the surface, positively and negatively charged amino acids, and it's reducing polar amino acids. So the model sort of find out that the best guess is to put on the surface charged amino acids and then don't think about the polars. If we compare this bias with Rosetta design bias, Rosetta has some issues that people knowing. So for example, for all residues, there's overrepresentation of alanines in the core. Sometimes the Rosetta is not able to pack things in the core, so it just puts alanine, things that are too close. There are too many tryptophans on the surface, and so on. So there are the different sort of biases that Rosetta is bringing in. Also, these models are sensitive to the backbone resolution. So whenever someone is reporting sequence recovery, you have to check what is the backbone resolution. So this is showing the sequence recovery as a function of the PDB resolution. And there's a clear trend for example, 3 angstrom resolution protein versus 1 or 1.5 angstrom resolution protein. Exactly the same story goes with AlphaFold model. So the more confident AlphaFold is about the structure, maybe it's closer to the crystal structure because it was trained on crystal structures, the higher sequence recovery ProteinMPNN can give. It was also important to predict uncertainty. So this is what I'm plotting is the sequence recovery at these 2 different temperatures, 0.1, 0.5. as a function of the negative log probability, so probability of a sequence giving a structure. And when we're designing something, we don't know the sequence recovery because we don't know what we're trying to get, but we get from the model the negative log probability of what the model designed. So we can still get an idea how confident the model is about this specific backbone and sequence match. So there's quite nice lines between the sequence recovery and the negative log probability. And then, for example, in this case, I try to design 32 sequences for these backbones and then rank them by the log probability. So the most probable sequence is this one, and then the least probable sequence is on the right. You see at the higher temperatures, there's only a very small sort of distance between the least likely sequence and the most likely sequence, whereas at these higher temperatures where there's maybe more randomness, there's a bigger gap between, for example, 48 and 50% sequence recovery. So one could sample many sequences with 50% or 0.5 temperature and then rank them by the score and pick the ones that satisfy specific features that they want. In this case, they will get more diversity. So now going back to the sequence, sort of this thinking about the backbone to sequence and sequence to backbone matching, what is the other way to sort of try to think, how can we evaluate, see how good the method is and what sort of people want they design a sequence? So one of the things that people do is this like forward folding. Okay, when I design the sequence, how would the sequence look? So in this case, AlphaFold, using AlphaFold in this single sequence regime, because often these sequences, these design sequences are quite different from the native sequences. So we're not even collecting multiple sequence alignments, it's just using a single sequence of AlphaFold to predict the structure and then check how well this input structure matches to the output structure matches to the input structure. So this is a structure, a model that we try to design. We come up with a sequence, and then we just want to double check how good the match is. So if we look at this native sequences, so 400 backbones, protein backbones, and take the native sequences without any homology data, the AlphaFold matching between the input and an output is really low because it's really hard to predict native sequences with just a single sequence without any evolutionary information. Those sequences are probably not very ideal. But then taking the same backbones and then redesigning a ProteinMPNN, they become way more idealized, more structurally ideal for the AlphaFold to predict. And we get a different distribution that a bunch of sequences can be repredicted to map the input. And similar idea, for example, for this de novo designed in TF2. So there's like a Rosetta design in TF2s, and then redesign the same backbones with ProteinMPNN. ProteinMPNN gives sequences that are more preferred compared with Rosetta sequences according to AlphaFold. So that's also good to see the match between the input and the output. So there's also, we trained with noise, so we can see how does the noise affect sequence recovery, and also how does it affect AlphaFold success rate. So that's quite interesting that adding this very small amount of noise in this PDB biunit regime reduces the sequence recovery from 55% to almost 51%. So this is what I'm talking about, the crystal artifacts of the PDB. But then as we increase the noise more and more, the sequence recovery slowly goes down. But what's interesting is that there's AlphaFold success rate. So for example, having the input and output match being 90 LDT C alpha or 95, it sort of increases as we're adding more noise. So it's also, the model is probably becoming more robust and giving more idealized sequences that could be easier predicted by AlphaFold. And it's also interesting that if we look at the similarities, if we take the same PDBs and sequences and generate a bunch of sequences and see how well those sequences match to this PSSM, so evolutionary generated similar sequences, that the model with more noise generates sequences that are more similar to the PSSM sequences. So we can think of this more of the consensus-like sequence. And I don't know whether it's just a coincidence or AlphaFold is actually better at predicting consensus-like sequences. Also, so when we think about this consistency check, the input backbone, and we have one model ProteinMPNN, and then we have a sequence and another model AlphaFold, the question is, when it doesn't match, why it doesn't match? Is ProteinMPNN came up with a bad sequence, or is it AlphaFold that it couldn't predict the structure? So this is showing this AlphaFold success rate to match these 90 and 95% cutoffs is the number of recycles. And you can see that having one recycle and 3 recycle being a baseline, so this 3.6% and 16% success rate. Increasing to 6 recycles, you get about 20% boost, and then increasing another 6 times recycles, you get another almost 20% boost. So it's sort of showing that the structure prediction is the hard part in this encoder-decoder regime. So often, even sequences that maybe couldn't be predicted by AlphaFold could be still folding into the correct backbones. So as I guess the structure prediction methods get better, there's more chance to sort of check yourself how well the input and output is matching. One of the last things also to talk about is that once we have this model that takes the backbone and produces distributions, we can think about combining these distributions. So in this case, I'm showing is that what if we want to design this multi-state backbone? So we want to sample a distribution that would, if we have either 2 chains or 2 states, that would, so I guess in this case, I'm showing this for homo-oligomer design. If I have chain A and chain B, and I want just one sequence, that would be A and B are exactly the same sequence. So I can predict the largest for both of the sequences, and then average them out to get this average blue distribution from which I can then sample. In more general, we can predict multiple backbones, either they're similar backbones or different backbones in different states. There could be, and then sum them up in some linear way, where the beta is some sort of a linear coefficient that is mixing these logics. And they could be positive or negative for positive design or negative design, multi-state design, coming up with a final sort of energy, we can think, and then making one distribution from which you sample and you populate all the states. And you do that again and again. So there's some interesting extensions of this. So some final points with ProteinMPNN is available on the GitHub. It's open source. It's fast and lightweight. There's almost no expert customization compared with Rosetta, except choosing which regions to redesign and picking some temperatures. It rescues a bunch of designs in the Baker lab. There is bias towards the surface charged amino acids, and maybe that's contributing to the high expression yields and solubility. You can do symmetric, negative, multi-state design, and so on. I'd like to thank all the people in the Baker lab that worked on this, and then we can maybe check the results from the AlphaFold forward folding, and then I can take some questions. So if we look at the, okay, we have 4 sequences, we can rank them by RMSD. So it's the best is 0.77 angstrom RMSD-C alpha. Those are the model scores, so how confident MPNN is about these sequences. Sequence recovery PLDDTs, and we can look that the structure is matching quite well to the input structure that we wanted to have. So it was quite an easy task, I guess, to redesign this protein. Yeah, I'm happy to take any questions.
Great, thank you so much, Justas. I think we can go over the questions in the order that they appear in the chat, so we can just
sure. So sounds good. Okay, so the first question is, can MPNN be used to improve a protein like mute compute does? I'm not entirely sure what mute compute does, but I think it is possible to use MPNN to make proteins more stable or like rescue specific things. Unless, I don't know, Giacomo, if you want to say more what mute compute does. Okay, otherwise I'll go to the next question from Kevin Yang. Does it still work if you don't use the RBF encoding for distances? Yes, it does. I haven't maybe used exactly just raw distances, but there are other encodings that can be used, or even discrete bins. I also try to use discrete bins just to get at this idea of not allowing the network to look at the too small of the distances, just discretizing, for example, distance at 0.5 angstroms or 0.2 angstroms. It does work in a very similar way. Can MPNN take multiple structures, conformations, and inputs? Yes, the MPNN can take, so it takes only one PDB file as an input, but you can input multiple backbones in the same PDB and just separate them in space. Since the model is a local graph sort of model, it will only pick on the nodes that are nearby, so it wouldn't pick on the other PDB that is far away. And that's how people, for example, in our lab are doing this multi-state design, just taking 2 states and inputting them in the same PDB and separating them apart. Do you input atoms other than amino acids like chemical ligands and metals? So in this version, it is not. Like I'm working on other versions that take in different metals, ligands, DNA, RNA, and so on. What's the theoretical or the best guess maximum accuracy? Presumably you don't expect to recover sequence exactly, given that many sequences are structurally homologous. I have no idea about the sequence recovery maximum accuracy. Most of the time, I usually look at the sequence recovery in the core, because I would expect that there's maybe more determinicity in the core, whereas the surface is harder maybe to recover, and it's also a function of the crystallization and so on. I have no idea what is the maximum accuracy. I guess I haven't emphasized, but in general, it's quite difficult to benchmark generative models. The same goes for, for example, the sequence design model. Like, how do you know when you come up with a better model? It's like better sequence recovery doesn't mean maybe that you have a better model. So having AlphaFold predictability really helps, but also probably setting up some large scale experiments would be a good idea at some point. Another question is, in natural language, random order models usually do worse than forward decoding. Do you have any intuition for why that's not the case here? One maybe answer is that there's not much data, so it's quite easy for the model to overfit, and maybe learning multiple decoding orders is preventing this overfitting. That's one answer. Another is maybe has something to do with this local sort of neighborhoods, that in this whole sequence is not a function of the left to right, it's these local neighborhoods that are determining, and they're not ordered, I guess, left to right. There's lots of this non-locality that also could do something with why that's also working. Another question from Simon is, for the ProteinMPNN plus fascialized protocol, do you think there is a biological reason why AlphaFold likes these structures better? Is there a difference between the different model noise levels and the AlphaFold PLDT of the ProteinMPNN plus fascialized protocol? I'm not sure about what are the PLDTs, so having certainty about the structure. What I was saying is that they were better predicted the line errors between the chains for the binder. Imagine the PLDTs maybe are also better. I don't know exactly what are these small changes that FastLX is doing to the structure that is helping for the MPNN to design a better structure and for AlphaFold to more confidently predict. Maybe it's idealizing some small bits, but yeah, I don't know. What was the ProteinMPNN train only on high resolution structures? Not sure if that's mentioned at the beginning of the talk. So it was not trained on only high resolution structures. I did try to train on structures that are, for example, better than 2 angstrom resolution, 2.5, 3.5 angstrom resolution. And the performances are quite similar. So I thought it's a better idea to train on more data, which means 3.5 angstrom resolution. Maybe it could get even higher, lower resolution. But I guess at some point it would start hurting. Also wondering if MPNN was trained on both crystal and cryo structures or just crystal. So it was only trained on crystal structures. We haven't trained on cryo-EM structures. It might be interesting to try. My feeling is that they might be slightly too low resolution. Another idea would be to actually use, for example, AlphaFold structures as an additional training set. Question from Livei Chang, would the model work for the multi-state design? If so, could you give an example of how robust the model for this case? It does work for multi-state design, we know. An example, I don't have data yet. I guess it'll be published at some point in the future. I believe the previous slide said 3.5 angstrom or lower. Okay, someone's responding. Is the code that integrates MPNN with FastRelax available? I think it should be available. I can ask Nate in our lab that set up the model. So ProteinMPNN is being incorporated into Rosetta as a C++ code that would be able to run from Rosetta straight away. So it should be made available, compatible. Have you thought about solution conditions and how to include that? I guess that might influence the surface side chain biases. I haven't thought about the solution conditions, but I did train a neural network without including any membrane proteins to try to have just the model to design soluble proteins. So maybe that's one of the ideas but I guess I will train a model to input maybe the outside conditions as whether it's a membrane or soluble protein as an input label. I guess that could be done maybe quite easily. How does the model handle different topologies of protein side chain connectivities like GFP, fluorophore? I don't have a specific answer for this question. I don't know, I have not tried, but we haven't seen any specific overfitting on different topologies or specific proteins. Can you sample different conformations of an interacting partner, say peptide, by starting with large noise level, temperature? How precise does that backbone have to be? So the model is not, I guess, sampling conformations, it's predicting a sequence. Maybe I misunderstood the question. But it would be possible, for example, to perturb the backbone, have some way to move the backbone, then sample a bunch of sequences and maybe repredict them to get an idea about the movement. Someone else is asking, what is the difference between options V_48002 and V_002 for modeling? So the models that I showed, I'll maybe just share again in the GitHub repo and also in here in this notebook. The models, so V is the version, 48 is 48 edges, so 48 nearest neighbors for the graph, and 0.02, it means there's 0.02 angstrom noise the model was trained with. This model was trained with 0.2 angstrom. This model was trained with 0.3 angstrom noise. So those are the models that are more robust to the larger noise levels. Okay, the next question is, regarding AlphaFold hallucination of the protein assemblies followed by ProteinMPNN sequence design, did ProteinMPNN improve the AlphaFold sequences mainly by making the surface more soluble, or was there evidence of important mutations in the core's binding interfaces? I would guess that it's mainly the surface redesign. There might be some core or like in between changes too, but I don't have statistics. We could ask Lucas and Basil and Alexei. Someone else asking, if I'm not mistaken, ProteinMPNN is deterministic. If so, have you experimented with decoding orders, a significant parameter to affect predictions? For instance, in interface design, does decoding from the residues proximal to the fixed sequence improve scores and vice versa? So I did try to experiment of initially, instead of using the random decoding order, try to design from the core to the surface, from the surface to the core, and other ideas. There's no clear evidence that it does help to go from some, I guess, geometrically inspired decoding order. It seemed like the performance is about the same. How long did training take, and what sort of hardware? So I trained on a 1 GPU, and I trained on A100 for probably 2 days. So the training is fairly fast. Do you think that B factor would be helpful input, perhaps for protein flexibility or disorder? It might be helpful, but at the same time, the real question is, what would be this input when someone is designing protein? Would they have an idea what sort of B factor to put in? Because most of the time, the model is used on some backbones that are either some sort of like helices that are parametrically designed, or AlphaFold backbones or some other backbones. So it's, yeah, I don't know whether it would be someone as a user would know what sort of B factor to use. How is MPNN performing with membrane proteins? I haven't tested that exactly, haven't looked exactly at the membrane proteins, but I do know that it does create membrane-like proteins if someone is trying to design something that is long and extended. I think ProteinMPNN thinks it's a membrane protein, so it puts hydrophobics on the surface. Another question is, ProteinMPNN score dependent on the decoding order? Yeah, are the log likelihoods from the model dependent on sequence context from the decoding previous residue? It does depend. The dependence is pretty small, but the score or log likelihood is a function of the decoding order. So if one wants to get a very precise score, can run MPNN multiple times and get an average and standard deviation to see what is the score. Usually the standard deviation is quite small. And final question is, what applications do you envision MPNN will be used for? So it's already used for different sort of redesigning sequences, trying to make them more soluble. I think there's some efforts in trying to use MPNN to make enzymes more stable. So for example, taking native enzymes and trying to redesign parts of them, trying to keep the function, but maybe make them more thermodynamically resistant, higher yields, and so on. So somehow trying to idealize making the proteins better. binder design is using MPNN, multi-state design, and all the other sort of sequence-related things. I guess an interesting result from this MPNN work was that a bunch of Rosetta designed things, ideal helices and so on, sometimes they wouldn't work, and probably mainly because the sequence was sort of incorrect or not ideal, that it was very hard to express or would oligomerize. So now this tool is helping to solve this question. If you have a decent backbone, can you come up with a sequence that work in experiments? But now I guess the hard task is now, how do you come up with backbones that you want to have? How do you come up with functional proteins and so on? Another question is, what would using amino acid encodings from the pre-trained protein language model as part of the input be helpful for training the model? It could be helpful. I don't know exactly. I guess yeah, one could try to do that. I haven't personally tried and looked into that sort of area yet. Any other questions? Another question is, if a backbone has more diversity at lower temperatures, could that mean it is more designable? So I would say if the backbone probably has less diversity at lower temperatures, the model is very certain about this thing, it would be maybe more designable. The model, it's probably makes sense to more look at the log probability to think about which backbones are designable. So how confident the model is about the backbones. So now asking, can you speak towards suggesting typical bias values or the bias by residue JSON flag? So there is a possibility to add biases. So for example, if we want more polar residues or other sort of, or maybe less alanines in the design. So these values are more just like experimental. I think there's an example in the GitHub repo that is suggesting some values, but otherwise probably some error and trial and error is needed to figure out what are those biases. I have not tried to design any CDRs for using MPNN, but I think it would be an interesting application. I imagine it's quite a difficult task, but it would be interesting to see. And I don't know whether training on specifically CDR loops would be helpful. So all the chains are included that are bigger than 30 residues, I think. So if it's in PDB, it's lower than 3.5 angstrom resolution, and bigger than 30 amino acids, it should be included. All right. Thanks, everyone. Does anyone have any more questions?
Thank you again, Justas. I'm going to stop the recording.